So in the previous sections, we have seen the pattern matching or exact pattern matching. So over there, what we were looking for was occurrence of some specific patterns within a sequence. So in this case here, we are given a pair of sequences, so that can be 2 DNAs or 2 proteins. And what we are trying to do over here is we are trying to compare both of them. So whole lengths of those 2 strands or sequences, they are compared with one another. So while we are doing this comparison, we were looking into different nucleotides one by one. So nucleotide 1 on the first strand compares to the nucleotide 1 on the second strand. So in this way, we are comparing a pair of nucleotides or amino acids. So that's why we call it as pairwise sequence alignment. In addition to that, we are having 2 sequences. So this kind of alignment is called as a pair sequence alignment. We want to compare the sequences to identify the regions of similarity or to find the conserved regions among them. What we can do with it? We can reveal similarities between the sequences, or we can determine the residue to residue correspondence in case of the proteins, and we can also locate the patterns of conservation. So we can locate some regions which are conserved between 2 sequences. We can study gene regulation with this, right, how the genes are regulated or controlled and how their expression is controlled. So for example, if we have some messenger RNAs which are produced by the DNA, so that is the when the gene expression takes place. But there are some elements like microRNAs, they go and block those genes. So in this way, we need to find the regions of similarity between this microRNA and its target messenger RNA, so we can identify what their targets are. So we can use this pair sequence alignment in this particular example where we can bind a microRNA to its target. We can also infer evolutionary relationship between the sequences, so how far they are and how distant they are. Let me introduce some vocabulary to you guys. So while we are comparing 2 sequences, we have sequence 1 at the top and sequence 2 at the bottom. We can look into those alignments in the shapes of the columns. So each nucleotide position, we can take it as column. So if we look into column number 1, we see there is an A in first strand and again an A on the second strand. So they are matching with one another. So in this situation, we call this is a match, whereas a mismatch is when a column contains 2 different non-blank residues, 2 different residues. So for example, here we have C and A. So in this case, we can call this is a mismatch. So indel is a column that contains a blank in it, blank because on one side we don't have a character. So in this case, we substitute with a dash like thing, whereas this dash, or there are many more present together, we call it as gap. So why we call it indel? Because we believe on one side there is an insertion, but on the other side there is a deletion. So the word indel comes from a phenomenon like this. So we see that again, match, where 2 characters match with one another, indel, where we have insertion and deletion together, and a continuous series of those blanks that can give us a gap. Even if it's just 1 character blank, we still call it as a gap. Mismatch is where we do have characters, they are not matching with one another. So there are 2 kinds of these pair sequence alignments. One is local alignments. So local alignments are where we can compare some small regions or sub-regions within those 2 sequences. So in that case, we call it as local alignments. And global is when we compare those sequences. In the alignment, we have both the sequences present, both full sequences present with their full lengths. Whereas in local alignment, we see that we have their parts of those 2 sequences, they are aligned together, and we can have more than 1 regions of local similarities. So here is how I can elaborate it again for you guys. So here is the alignment of 2 subregions. So these 2 sequences, they align here, don't align here, alignment here, no alignment here. So this kind of arrangement is called as the local alignment. Local alignment is appropriate when aligning 2 segments of DNA that may have local regions of similarity which are embedded in a background of non-homologous sequences. Whereas the global alignment is in this sequence alignment over the entire length of 2 or more nucleic acids, we will do that in later lectures. We'll talk about multiple sequence alignments. So both the terms, they are used over there also. So whenever we say global, it means the sequences are aligned end to end. When we see local, they are where we have some local regions of similarity. So in a global alignment, the sequences are assumed to be homologous along their entire length. So in this section, we have just introduced the pairwise sequence alignment, and we also look into what is the difference between local alignment and a global alignment.